You've now been introduced to the evolution of skeletal systems in the vertebrates, and you've learned a little bit about the properties of skeletal systems. And so now we're going to start looking at the different components of the vertebrate skeletal system, beginning with the cranial skeleton, otherwise known as the skull. In this web lecture, I'm going to present the organization, the development, and also the evolutionary history of the cranial skeleton. Your book presents a couple of different kinds of ways to organize the vertebrate skeleton. The first one is dividing it into an exoskeleton and an endoskeleton, with the exoskeleton being composed of the dermal bone on the external part of the body and keratinized structures from the epidermis on the outside of the body, and the endoskeleton being composed of bony cartilaginous skeletal elements internal to the body, as well as the notochord. Another possible way of dividing up the skeleton, which is the organizational scheme we're primarily going to be using, is first dividing it by cranial skeleton versus postcranial skeleton. So cranial skeleton is the skull, postcranial skeleton is everything other than the skull. The postcranial skeleton can be divided into the axial skeleton, which consists of all of the midline structures of the skeleton, the vertebral column and ribs primarily, but a few other structures that we'll go into when we get to the axial skeleton, and the appendicular skeleton. So these are basically the limb bones or fin bones if you're talking about fish. For the cranial skeleton, it's divided into three major divisions. So the first is the Contra Cranium, also known as the endocranium or the neurocranium. And the job of the Contra Cranium is to surround and protect the brain and the major sensory organs, so the eyes, the ears, and the nose. The Splanchnic Cranium consists of the components of the skull that are derived from branchial arches. So you'll remember back to those pharyngeal slits that we saw all the way going back to the beginning of the deuterostomes. We saw that we have some skeletal elements that support those pharyngeal slits, and these become the branchial arches. We'll talk much more about the evolution of the Splanchnic Cranium as we go through this web lecture. And finally, there's the dermatocranium. So this is the dermal bone component of the skull, and it covers the majority of the skull in most vertebrates. So it's going to be that kind of protective exoskeleton part of the skull. So let's begin by discussing the chondrocranium, and we'll begin with the development of the chondrocranium. So in the very earliest stages of development of the chondrocranium, it begins first with some small little spicules of cartilage. This one is called the trabecula, and then a pair of paracortals that are going alongside of the notochord you see protruding up into the cranial area, as well as some little condensations of cartilage that are going to become the occipital bones at the very posterior part of the skull. And then the other very early thing to develop are the sensory capsules. So we have the nasal capsule, which is going to form the nose, the optic capsules, which are going to be the eyes, and then the otic capsules, which are going to surround the ears, the hearing and balance apparatus. As development proceeds, these little spicules of cartilage expand out into sort of a flat plate. These occipital little condensations are going to grow together and form an arch that surrounds the notochord and later, of course, the neural tube. At this point, these spicules are just going to form sort of a base underneath the developing brain. So think of it as kind of like a bed for the brain at this point in development. Again, the sensory capsules develop very early and they continue to develop at this stage. And then finally, in the later stages of development, that sort of base for the brain, that little bed for the brain, grows up and around invertebrates with an ossified skeleton. So the osteichthyes, this is going to ossify at this point to create a whole protective case around the brain. And so the parts of this chondrocranium I would like you to know about are the ethmoid, which surrounds the nasal capsule, the sphenoid, which surrounds the anterior part of the brain, and the occipitals, which surround the posterior part of the brain, as well as the sensory capsules, nasal, optic, and otic. So just to review this in an adult version of this chondrocranium, the chondrocranium consists of nasal and otic capsules. So we've got the nasal capsule here anteriorly, the otic capsule laterally. The optic capsule actually does not join with the cranium, so the optic capsule becomes the sclera around the outside of the eyeballs so that it can move independently of the skull. It doesn't actually become fused with the skull. And there's an ethmoid region anteriorly again around the brain and adjacent to the nasal capsule. Centrally, we've got the sphenoid region that's going to surround and protect the middle part of the brain, and then an occipital region in the farthest posterior, so you can see this whole series of different components of the occipital bones that surround this hole called the foramen magnum, which is the exit point for the nerve cord. Let's now turn to our second major division of the cranial skeleton, the Splanchnic Cranium. So when we're talking about the Splanchnic Cranium, we're talking primarily about jaws. So this is one of the major transitions in vertebrate history, the origin of jaws, and so that's associated with this group called the gnathostomes, boxed in here. So that includes the cartilaginous fishes, the chondrichthyes, plus all the osteichthyes. Those are the two main radiations of gnathostomes, and gnathostomes are defined as jawed vertebrates. That's one of the primary synapomorphies. This is also accompanied by a number of other synapomorphies. So the number of pharyngeal slits is stabilized to 5 within the gnathostomes. We begin to see jointed visceral arches, so these skeletal supports that support those pharyngeal slits that we've talked quite a bit about already are now supported by these jointed visceral arches. We also begin to see paired lateral appendages, so pectoral fins and pelvic fins in the gnathostomes. Paired nostrils, so lampreys and hagfish have only a single nostril. Now we begin to see 2. And also the presence of 3 rather than 2 semicircular canals. So these are the little balance organs in the inner ear that give you information about rotations of your head, where you're moving in space. So this is an indication that these gnathostomes were beginning to make more use of the vertical water column in the oceans rather than being primarily bottom dwellers. They needed to have information about the vertical dimension as well as the 2 horizontal dimensions, indicated by that 3rd semicircular canal. And the origin of gnathostomes is also associated with another doubling of these Hox genes, most likely indicating another whole genome duplication because both of the 2 sets of Hox genes were doubled for a total of 4. So if we look at the phylogeny, the outgroup, which would be something like a fruit fly, the protostomes, we see 1 Hox cluster. In the cephalochordates, we also see 1 Hox cluster. Here we have this first Hox gene cluster duplication, and in lampreys we see the 2 copies of all the Hox genes. And then when we get to the gnathostomes, we see another doubling of both of those 2 sets of Hox genes for a total of 4. Now let's take a look at the evolution of these jaws. And as I said before, the story begins with pharyngeal slits. When we first encountered pharyngeal slits, we were looking at them in non-vertebrate chordate organisms such as this landslit, also known as Amphioxus, where we saw that the primary function of them was in suspension feeding. So if you recall, we have a flow of water in through the mouth. It's going to come into this area called the pharynx. In the floor of the pharynx, there's this organ called an endostyle that's secreting a sticky mucus. The food particles get trapped in the mucus and carried into the gut, and the water is forced out through these pharyngeal slits after the food has been removed. So again, water in through the mouth, out through the pharyngeal slits. And this flow of water was created by the action of cilia, little hair-like cells that wave to create a current of water. So this current of water is not going to be very fast. It's not going to have a lot of force behind it. It's not going to move very large food particles, just very small food particles that are suspended in the water. We can think of the evolution of the jaws from this point as a series of stages that will serve as pre-adaptations for the later evolution of the definitive jaws. And so remember, when I say a pre-adaptation, what I mean is structures that evolved in an ancestor for some purpose other than the definitive purpose that were later able to be co-opted or incorporated in a new structure with a new function. And that new structure and function would not have been possible if that structure had not already evolved for some other purpose. What I do not mean when I say a pre-adaptation is that there was some sort of anticipation on the part of evolution of some later need. Think of it as a happy accident. It was there for another purpose, and it is just fortuitous that it's useful for some different function later on. So we can think of the first phase as the one that we were just looking at, this chordate ancestor that moved water by ciliary action, so the waving of cilia creating a current of water. And in this chordate ancestor, as in living non-vertebrate chordates, these pharyngeal slits are supported and held open by collagen structures. In the second phase, we can think of as early vertebrates, we start to see constrictor muscles surrounding the pharynx. So now the pharynx can work as a muscular pump to move water through. The muscles are going to contract and squeeze and decrease the volume of the pharynx, and that will push the water out through the pharyngeal slits. And when they relax and open up again, that's going to increase the volume of the pharynx and draw water in through the mouth. So to be able to do that, you need some stronger skeletal support, because remember, muscles only pull, they don't push, right? They can only shorten. They can't push something back open again. So you need those skeletal supports to open them back up again. So now we start to see these pharyngeal slits supported by cartilage arches that give it some elasticity to regain the original volume. In the next stage, we start to see these pharyngeal arches, also known as branchial arches, in a jointed form. So there's now sort of a hinge action to them, and muscles are going to be attached to these jointed arches so that they can be actively controlled to change the size and shape of that pharyngeal cavity more actively and more precisely. At this point, the water flow was most likely being used for respiration. Remember, gills are a synapomorphy of the vertebrates, and so at this point we're using this flow of water to ventilate the gills and not so much for suspension feeding. In the final stage, the anterior-most of these pharyngeal arches, also known as branchial arches, join together in the front and fuse together to form the familiar jaw structure that we see. The musculature is already in place, so remember, we see muscles associated with these jointed branchial arches. That muscle is carried straight over into the jaw, so we already have musculature to allow that jaw to open and close like a hinge. Most likely, this was still being used primarily for respiration rather than for grabbing food the way we see jaws used now. Now let's take a closer look at the anatomical structures involved. Here we see a cutaway view of the jawless condition, so we would call this the pre-gnathostome state. Each of these little arches you see is a skeletal structure. We see them in the jointed form. And so we can see we have 1, 2, 3, 4, 5, 6, 7 branchial arches at this point, with a couple of arches that we are calling premandibular arches. So these are anterior to the arch that's eventually going to become the jaw or the mandible. And you saw some discussion in your book about competing hypotheses about what actually becomes of these premandibular arches, both evolutionarily and developmentally. One hypothesis states that they both just become incorporated into the brain case above. The other hypothesis suggests that some components of it are incorporated into the brain case and some are actually incorporated into the definitive jaw. So here we see these jointed structures. This orange one is going to become the mandibular arch. will eventually become the jaws. The second arch is known as the hyoid arch. The third is known as the carotid arch. We're going to see a lot of this terminology over and over and over again when we get into the circulatory system and the nervous system. And then the rest of these arches are known as vagal arches. They're not differentiated. So when we go to the early jawed condition, now we see, in this case, it's showing these premandibular arches incorporated into the base of the neurocranium or the Contra Cranium. These mandibular arches joining at the front to form the jaws. The upper part of it, in its cartilage form, is known as the palatoquadrate cartilage. The lower jaw, in the cartilage form, is known either as the mandibular cartilage or also as Meckel's cartilage. And again, we've got the hyoid arch and the carotid arch, and then the vagal arches behind. Finally, we see this hyoid arch modified to help support the jaws. We'll talk much more about that later in this web lecture. And then the rest of these arches continuing to function as support for the gills and for the constrictor muscles around the pharyngeal slits. Now let's think some more about this second arch, the hyoid arch shown here in blue, and what it ends up doing as part of this whole jaw apparatus. So one thing that we can imagine when the jaws first formed is that they're basically surrounded by muscle, kind of embedded in soft tissue, and so they can open and close, but they don't really have anything to transmit force. So as we know from physics, anytime you try to apply a force to something, it's going to apply a force back. If you can't resist that force, then basically you've done nothing but just move yourself around. So we can think of this as these kind of disembodied jaws that can't really exert any force because they're just going to be shifted around within the mouth unless they have something to brace them and to push against to be able to transmit that force. So this becomes the purpose of the second hyoid arch. So this is going to form a structure called the Hyomandibula that is ultimately going to create that brace between the jaws and the cranium that's going to allow the jawed vertebrate to transmit the forces from its jaw to be able to have a more powerful bite. As we said before, the rest of these arches, which remember is stabilized to 5 within the gnathostomes, so 1, 2, 3, 4, 5 branchial arches accompanied by 1, 2, 3, 4, 5 gills in between them, are going to continue to have that function of supporting and protecting the gills, and they're divided into a different series. So you have seen this in your shark dissection. So there's a basibranchial, here most ventral, hypobranchial, ceratobranchial, epibranchial, and pharyngobranchial series of these branchial arches. The ventral part of the hyoid arch becomes the hyoid apparatus that helps to support the tongue and the floor of the mouth, and it retains the same general structure as the branchial arches. There's a basohyal, hypohyal, and ceratohyal. The dorsal part of this becomes the hyomandibula. So let's take a look at how that works in helping to support the jaw. So in the earliest condition, which is known as primitive autostylate, the palatoquadrate is braced directly against the base of the skull without any involvement of the hyomandibula in transmitting those forces. So the mandibular cartilage will push up against the palatoquadrate, which will in turn push up against the Contra Cranium. So this is the primitive autostylic condition, also called in your book the euautostylic suspension of the jaw. So jaw suspension refers to the bracing of the jaw against some other structure to be able to resist forces. So this primitive autostylic suspension leads to a more advanced form of suspension known as amphistylic. So in this form of suspension, the hyomandibula makes a connection both with the Contra Cranium and also with that palatoquadrate, the upper jaw, to have a very, very stable form of jaw articulation. So the jaw is supported against both the Contra Cranium and also is being braced by that Hyomandibula. So this form of jaw suspension gives rise to two different, later, more derived kinds of jaw suspension. One is known as hyostylic suspension. So in hyostylic suspension, the upper jaw, the palatoquadrate, has lost its connection with the base of the cranium and is supported entirely by the Hyomandibula. So this has a very great advantage because this Hyomandibula is mobile. It will pivot around the Contra Cranium. So this allows fish with this kind of jaw suspension to have a form of cranial kinesis that allows the jaws to be protruded forward. And so in images like this with a shark, you can see that the mouth is kind of back underneath the nose, and then when it deploys this hyostylic jaw, the jaws actually swing forward. So here you're seeing kind of an internal view. Here's the Hyomandibula, the ceratohyal, and the jaws attached to it, and this Hyomandibula apparatus just pushes the jaws entirely forward. So when you see images like this from jaws where it looks like those jaws are coming straight out of its mouth toward you, it's because in fact they are. The other form that's more derived is a secondary form of autostylic suspension. So remember, autostylic means that the upper jaw is attached directly to the base of the cranium, and so now there's no longer a need for this attachment to the Hyomandibula, and we see the primary bracing of the jaw being between the jaw and the base of the cranium. So we see this in some of the cartilaginous fishes, the holocephalans, as well as lungfish and tetrapods have this form of jaw suspension. We will continue the story of the Splanchnic Cranium, but before we do, we need to understand a little bit about the dermatocranium. This is the dermal bone component of the skull, so this is the exoskeleton part, and it has its origin in the external body armor we've talked about with reference to the early jawless fishes. So some examples of them are shown here, and they're covered by this protective armor, sometimes covering the whole body and sometimes restricted mostly to the head. And the dermal bones of the skull, the dermatocranium, are the remaining elements from this external body armor that continued from this early radiation of jawless fishes into the jawed fishes, such as the placoderms shown here, that retain this covering of external body armor in the form of dermal bone. And what happens developmentally is that this dermal bone actually overlays and covers up nearly all of the endochondral bone of the Contra Cranium and Splanchnic Cranium. And so here we see the Splanchnic Cranium and Contra Cranium here in yellow and blue being covered over ultimately by all dermatocranium shown here in red. And here you can see this sort of see-through view of the dermatocranium just covering up all of those chondrocranial and splanchnicranial elements underneath to form the head of the fish. And this happens in series of small individual bones. And so these series are formulated a little bit differently from what you see in your book, but it covers all the same basic bones. So we have a mandibular series that covers the lower jaw. We've got a gular series that covers the floor of the mouth. There's an opercular series that is the bony covering for the gills. So one of the synapomorphies of osteichthyes is having a bony operculum covering the gills. There is a dermal roof that covers the top and sides of the cranium, and then a palatal series, which you can't see, but it's covering the inside of the top of the roof of the mouth. And the fact that we have all of these individual small articulating bones creates the possibility of cranial kinesis. So what we end up in more derived, particularly teleost fish, is this highly mobile dermatocranium that allows for very elaborate jaw protrusion. We'll see a very dramatic example of this in class. And it's operated by that 4-bar linkage system that you read about in your book. So these large arrows represent the muscle forces that are applied to the different linkages in that 4-bar linkage. And so when all of these muscles are deployed, the maxilla and premaxilla are sort of thrown forward, similar to what we saw in the shark. Only in the case of the shark, it was the Splanchnic Cranium that was driving that cranial kinesis. In the case of these fish, it's the dermatocranium that allows this extreme jaw protrusion. Cranial kinesis continues to be present in many groups of tetrapods, most notably the squamates, the snakes and lizards. The skull you're looking at right now is the skull of Tuatara, which is a lepidosaur, but not a squamate, so a close relative of the snakes and lizards. And remember that lepidosaurs are diapsids, and the diapsid condition is related to the presence of these sort of holes or windows in the dermatocranium. So these are called temporal fenestrae. So they have one here, a lower temporal fenestra and an upper temporal fenestra, and they're very easily identifiable in the Tuatara. You may remember the first week of lab, you saw a Tuatara skull as part of the skull exercise. And cranial kinesis in squamates is characterized by extreme reduction in the dermatocranium. So in snakes and lizards, what we see is the loss of that lower temporal bar. So that lower temporal fenestra, so fenestra literally means window, so we can think of this as kind of a window in the dermatocranium. That lower temporal fenestra is no longer even recognizable because it's lost its lower border. And the bone that makes that up, that the quadratojugal bone that defined that lower temporal bar, has been lost in this modern lizard. And we see even greater reduction of the dermatocranium when we look at modern snakes. So we can see this sort of progression of decrease in those dermatocranial elements and an increase in skull kinesis as we go through different groups of snakes and lizards, starting with the Tuatara, which remember is outside that group. Chameleons have some cranial kinesis, but you start to see more and more as you get into things like the gecko, and very high cranial mobility in snakes. This will be familiar to most people as this idea that snakes can unhinge their jaws to swallow prey that's much larger than the size of their head. They actually don't unhinge their jaws, but their entire jaw mechanism and their skull is incredibly flexible because of this reduction in dermal bone. So cranial kinesis in snakes is accomplished through a series of 8 jointed links on each side. So these bones are kind of reduced to just bars, basically, that can move relative to each other. And each side of the jaw moves independently. So the mandibles, the lower jaw, are joined in the center only by soft tissue, by muscles and skin. So they can actually, the two sides of the jaw can spread apart from each other in the center midline. So the way they do this when they're swallowing prey that's very large is that they'll use one side of their jaw to hold the prey item in place, while the other side is going to stretch and advance over the top of it. Then that side grabs on with its teeth, and the other side releases and kind of walks over the prey to move it down into its throat until it's completely swallowed. Now that we've learned a little bit about the dermatocranium, we can come back to the story of the Splanchnic Cranium. So remember that the Splanchnic Cranium is the part of the skull derived from branchial arches. So it includes the elements derived from the first and second arch, the mandibular and the hyoid arch, but not the other branchial arches. So we saw that in their cartilaginous forms, they are known as the palatoquadrate, and that's the upper jaw, and the mandibular or Meckel's cartilage, that forms the lower jaw. And those are the definitive jaw in chondrichthyans, so cartilaginous fish, continue to have those structures. And then the hyomandibula is, of course, also part of the Splanchnic Cranium. During ossification, the Contra Cranium, remember we just said, is totally covered over by dermal bone. But the Splanchnic Cranium is almost completely replaced. It's not just covered over, but it actually replaces most of the bone of the Splanchnic Cranium. So when this happens, there are two little bits of the original palatoquadrate and mandibular cartilage that survives this ossification process. So the palatoquadrate ossifies as the quadrate, which most of the time is a small little bone at the very back of the jaw that forms the articulation between the upper and lower jaw. What's left of the Meckel's cartilage is this ossification called the articular, which forms the lower part of the jaw articulation. So the hyomandibula and the hyoid apparatus ossify as their own structural elements and are not replaced by dermal bone. So we can see this in various animals. These are choanate fish. We see a number of dermal bones that replace that lower jaw. The tooth-bearing bone in the lower jaw is called the dentary, but there's a whole series of other bones in choanate fish that form the lower jaw. So splenial bones, the angular, the surangular. This is looking at the outside view. The internal view of the lower jaw. We see the prearticular, a little bit of the angular from the other side, and here is that articular bone, that little remnant from the Splanchnic Cranium that forms the jaw joint. In tetrapods, we see the same thing. Lots of separate bones that make up the lower jaw. The dentary is always the tooth-bearing bone in the lower jaw, and a whole series of different bones. And then if we look again at the internal view, the articular is the one little remnant left of the Splanchnic Cranium. Same situation in the upper jaw. So now we're looking down at the top of the skull of an early tetrapod. And again, a whole series of different dermal bones that cover over that Contra Cranium. And in the very, very back, we see this little remnant called the quadrate that's going to form the upper part of that jaw joint. And this is a palatal view, so you're looking up into the roof of the mouth here. The tooth-bearing bones in the upper jaw are the maxilla and premaxilla. They're the main tooth-bearing bones. And then we can see the quadrate in the rear of the mouth forming the joint. When we get to tetrapods, remember that we see the hyomandibula in the secondary autostylic configuration. So we've got secondary autostylic jaw suspension, or what your book calls metaautostylic jaw suspension, where once again the upper jaw is fused directly to the base of the skull. And the picture that you saw before of this when we looked at all the different kinds of jaw suspension was a little bit misleading. It showed that the hyomandibula was sort of sticking out to the back like this. But in fact, in tetrapods, the hyomandibula retains that ancestral connection to both the cranium and the upper jaw. So in secondary autostylic suspension, the hyomandibula is positioned basically between the upper jaw and the region of the skull where the ear is. So there's no longer any need for the hyomandibula in jaw suspension. The jaw is directly braced against the skull in tetrapods, but it happens to be perfectly situated to transmit sound from the jaw directly to the ear, and thereby amplifying the sounds and improving hearing. So at this point, the hyomandibula in tetrapods becomes known as the columella. So here we see the columella right there, diagrammed in purple in tetrapods. This is an early tetrapod that you're looking at here. The hyomandibula becomes the columella and takes on this new role in the transmission of airborne sounds. So it retains this ancestral connection between the quadrate here, shown in green, and the temporal region of the Contra Cranium here in blue. And this arrangement of bones is retained in living tetrapods in the sauropsid lineage. So here we have again the tuatara, and again you can see the quadrate and the articular forming the jaw joint. In tetrapods, the palatoquadrate ossifies not only as a quadrate but also as this bone known as the epipterygoid that you see running through the middle of the skull. And here's another example of the tegu lizard. Again, the articular and the quadrate form the jaw joint, and we see this other ossification of the Splanchnic Cranium known as the epipterygoid. The columella functions still in these groups in sound transmission between the jaw and the inner ear. So we can see another couple of examples in diagrammatic form. This is this amphibian, so a modern amphibian such as a frog. So here we have the articular forming the lower jaw joint, the quadrate, and then followed by the columella, which you see spanning from the eardrum, the tympanum, to the middle ear cavity. Same thing in a lepidosaur, such as a snake or lizard. Again, articular, quadrate, columella in that order. They form a chain of bones in the same order, and then the columella still plays that role in sound transmission, again from the eardrum to the middle ear. Here are another couple of examples showing this columella, again spanning between the eardrum, the tympanum, and going to the middle ear. And in the case with snakes, you see this columella becomes very, very long and slender, attached to the quadrate, which forms that jaw joint. And in atympanic snakes, so snakes who have lost this external eardrum, like you see here in the squamate, in the lizard, this is basically their primary means of hearing. They sense vibrations through the ground as they slither along through their jaw joint, which is transmitted through the quadrate and columella right into their ear. So this is their only mechanism for detecting primarily very low frequency and high amplitude sounds. And so remember that in the case of squamates, we saw this very dramatic reduction in the dermatocranium, the reduction in the number and the size of the different dermal elements in the skull. In mammals, we see exactly the opposite trend. So in the evolutionary history of mammals, there was an increase in the reliance on intraoral processing of food, basically chewing, mastication, to facilitate more efficient digestion in this group. Mastication is associated with such morphological features as heterodonty, having different shaped teeth in your mouth, and also the formation of muscular cheeks to provide the muscle for chewing. And this represents, in the case of mammals, this trade-off between stability and mobility. They have very, very akinetic skulls, very strong and stable skulls to be able to withstand the repeated forces inherent in these chewing motions. So bone articulations are weak spots when you're undergoing a lot of repetitive force. So what we see in mammals is an increase in size and a decrease in the number of the different dermal skeleton elements. And one of the most striking is this increase in size of the dentary. Remember, the dentary is the tooth-bearing bone in the lower jaw. And so through evolutionary time, we're seeing a series of different synapsids leading up toward mammals. The dentary becomes larger. These other bones that made up the lower jaw in other tetrapods become smaller and smaller as the dentary takes up more and more of this lower jaw, until finally, in the end, the dentary is the only bone in the lower jaw of mammals. And again, this is an adaptation for strength and stability in that lower jawbone. And we see this throughout the mammalian skull. So this is an example of a cat skull, and all of the bones are very highly fused. The skull forms this really nice solid dermal bone shield that protects the entire head and provides that stability needed with chewing. So drastic reduction in the number of bones and articulations in the skull in mammals. The bones are highly fused for a very strongly supported head, capable of withstanding chewing forces. So what happened to the rest of those bones? So Early on in mammalian evolution, the dentary is still the main tooth-bearing bone, but we still see other bones such as the angular, superangular, and of course the articular and quadrate early in mammalian history still formed that jaw joint. So we still have this association in early mammals of the columella with the quadrate, so forming that chain of bones that we saw in many of the other tetrapod species, articular, quadrate, and then columella. You can't see it in this image, but it's in there. So as the dentary enlarges, the other bones of the jaw become reduced. So we see the angular, articular, and quadrate. Eventually, the dentary becomes so large that it gains a secondary articulation with the squamosal bone, also known as the temporal bone. So at this point, the early mammals had a double jaw joint. They had both an articulation between the dentary and the squamosal bone, and also between the quadrate and articular. So this is the beginning of the double jaw joint. When the dentary takes over the entire lower jaw, only small remnants of the other bones are going to remain. So the quadrate and articular are becoming very, very small now. This other joint between the dentary and the squamosal is going to be strengthened and is going to be much more the force-bearing joint that the mammals are using to withstand those chewing forces. And so as these bones that used to form the jaw joint lose that function, they become more weakly articulated and become very, very small in size, which makes them very, very adept at vibrating in response to airborne sounds, even fairly high frequency and low volume sounds, low amplitude sounds, are going to cause these to vibrate. And remember, we still retain that ancestral connection between the articular, quadrate, and columella. So here we see in a primitive teleost, articular, quadrate, Hyomandibula, making that connection with the region of the ear. So the columella, remember, is connected to the quadrate due to its earlier role in jaw suspension. So we've got this chain still, articular, quadrate, columella. So the quadrate is still connected to the articular due to that earlier role in jaw articulation. And they retain these connections, but all three of these bones migrate into this middle ear cavity to form the ear ossicles, the small ear bones. So here we have the articular, which is now the one that's making the connection with the tympanum, or the eardrum. So this is now known as the malleus. The quadrate is now known as the incus, and the columella is now known as the stapes, and this makes that connection with the internal membrane to transmit sounds from the middle ear to the inner ear. And so we see that in these very early true mammals. This is a synapomorphy of the mammals. Again, malleus, incus, stapes being this chain of tiny little bones that are used for amplifying sounds in mammals. And we still also retain a remnant of one of those other lower jaw bones, the angular, which becomes the ectotympanic bone. Ectotympanic bone that surrounds and supports the tympanic membrane. So, most important points from this web lecture. The functions of the Contra Cranium are protection of the brain and sensory organs. We saw the evolution of jaws from pharyngeal arches through a series of pre-adaptations. Make sure you're familiar with those. And also the earlier functions of all of these structures that eventually led to the evolution of jaws. We saw that the skull is a dual structure that shows a trade-off between stability for protection and mobility for feeding. So we saw lots of feeding adaptations that emphasized either mobility or stability in different cases. We saw that cranial kinesis, or extreme mobility of the skull, is accomplished by the Splanchnic Cranium through jaw suspension. We saw this in the sharks with that hyostylic jaw suspension, and by the Dermatocranium through having a large number of articulating parts, such as the case in the more derived fish and the squamates. And then we saw the further evolution of the Splanchnic Cranium from being jaw articulation and jaw suspension to having a role in the transmission of sound for hearing.